Hi everyone! In this video I'll be talking you through the sectioning of an embryo, what you'll see, and how it translates into an embryo's 3D shape. Here's our transparent embryo, if you will, where I'm going to put the shapes of various organs as they sit in the living embryo. And here's our embryo for sectioning. If you want to follow this at home, I'm using salt and flour dough, which you can make at home, and just a wee bit of plasticine for the colourful bits. An embryo at this stage, the end of week 4, is kind of curved over like a kidney bean. But when you're given a histological slide of an embryo, it's been cut in these very, very straight parallel lines to produce those slides. This can make it a wee bit harder to orient yourself, because the slides don't map neatly onto the concepts of ventral, dorsal, rostral, caudal. Let's take a few more sections here. So what would we see looking down the microscope at sections taken along these lines? These slices of our model will be our slides, and I will lay the structures on them as I go through. Five slides for you. The neural tube is one of the easiest structures to see on an embryo. It's made from a groove in the ectoderm which closes off into a tube. So what we see on the slide is this white lumen surrounded by sturdy darker cells. There's our neural tube. It bends like this on the dorsal side of the embryo. Now what will it look like on the slides? Let's cut it. Now our neural tube is sectioned off. Down in this section and up here it looks like there are two neural tubes because it curves around and the sectioning has caught both ends of this curve here. Up at the rostral end we're seeing a lot of this neural tube tissue, and this is our primitive brain. It's been cut lengthways there. Let's go to an endoderm-derived structure next. By the end of week 4, the gut tube has separated into the foregut up here, the midgut in here, and hindgut down here. The yolk sac is still present. It's up by the umbilical cord here. Here is our yolk stalk. This bit here is the allantois, the bit of the gut tube that goes into the placenta. Let's cut these and put them onto our sections. On slides, you'll see the gut tube as this little ring in the midline of the body, usually in the middle of a septum. One place where that's not true is places where the gut comes out, like the yolk sac here, or the allantois here. Now for the heart, which is this little bulge in purple here. At this stage, the heart has folded over, but septation hasn't occurred yet. On these two slides, we're going to see the start and end of a muscular structure with wide spaces in it, which are our primitive heart chambers. And there we have some slices of heart at the rostral and caudal ends of the heart. Next up, we've got the liver, which is another big landmark. It's really oversized at this stage. It bulges out a lot. It fits in the abdominal cavity here. And on the slide, you see it as a big, more solid mass that takes up a lot of the abdominal cavity space. Let's put it in. That's our liver section, nice and wide, nice and big, in our abdominal cross-section of the embryo. Now the last thing I'm going to show you here is the mesonephroi, these paired embryonic kidneys, which then become the urogenital system. They sit round about here I made one red and one orange so that you can see that they are paired. Let's cut them and see where they go. On slides, these appear as these kind of irregular patches, and they will also have kind of white holes in them. And that is my sectioned embryo, all done. Here are the structures in place, and as they appear on the microscope slide, if you section them like this. The same ideas can be applied if you're looking at an embryo that's been cut in another plane, like down here, or maybe obliquely across here. You just need to know what you're looking for. Thanks everyone.